Another paper, I wanted to highlight one particular section on this. This paper, 3 subsets of sequence complexity and their relevance to biopolymeric information. They're refocusing on DNA, RNA, and proteins. They point out there are 3 types of complexity. Random sequence complexity, where the laws of nature do not determine the exact outcome, like I explained. There's ordered sequence complexity, where the laws of nature do determine the outcome very rigidly. For example, the structure of a crystal lattice is very ordered. But there's a 3rd type of complexity, and neither of the first 2 are what we're finding in the biopolymers. The 3rd type is functional sequence complexity. And functional sequence complexity, they discuss at length. There's only 3 places we find it: human languages, computer code, and the information encoded in the genomes of life. Those are the only 3 places we're finding functional sequence complexity. But note what he says here: without volitional agency assigning meaning to each configurable switch position symbol, algorithmic function and language will not occur. Now, I realize that's just one sentence taken out of the paper. You can actually, I've actually published a paper afterward, reading this paper with these guys. Because in that paper they didn't see how you could possibly measure functional sequence complexity. I was working on that problem already. I contacted them. They asked a series of questions. Well, what about this, what about that? And then we went on to publish another paper on how to actually measure this stuff. The same year it was published, another paper was published by Hazen and Zostak and a couple of other fellows who also proposed a way to measure functional information, which in our paper we said is identical to functional complexity, functional information. Zostak, I think it was 2003 in Nature, said, look, we have a problem in genetics. The problem is that the information that we see in genetics is different from the kind of information they worry about in information theory, where it doesn't really matter whether it contains any meaningful or functional stuff. It can be pure gibberish. They're just more worried about the transmission of bits and bytes from A to B. He says in genetics it matters very much whether that information is useful or functional. So he proposed a new term called functional information. And then a few years later, in 2007, they published a paper on how to measure that, a simple equation. We published a paper that same year, a little more sophisticated, that you can actually use for proteins. If you simplify it, you actually get their simpler equation. So we're both on the same track. That is a good thing. When 2 people come at a problem from 2 different angles and begin to converge on the same solution. Volitional agency is the phrase I wanted to point out here. First time I've ever actually seen that in a peer-reviewed science paper, where they're coming right out, these authors say, we need intelligence to write code, basically, and the code that we're seeing in the biopolymers doesn't occur without intelligence. Choosing the configurable switch position symbols. Here's a hypothesis I want to bounce off you. I want you to think about this. It might take a few weeks, months, or maybe even a few years to digest. You might resist it a lot at first, but it's worth thinking about because I'm just not sure there's any way out. It's the functional information hypothesis. A unique property of intelligence is the ability to produce statistically significant levels of functional information. That's an hypothesis. But, key word is unique. The only place, if you see functional information, if it's unique to intelligence, it's like the fingerprint or a fingerprint of intelligence. So if you see an effect, or you get a signal from deep space that has a high degree of functional information, that is the fingerprint of intelligence. You know it came from an intelligent source. Keep that in mind, because what that does is it provides us with a scientific method for design detection, the detection of intelligent tampering that we can use all over the place, not just in forensic science and biology, but in SETI, in archaeology, and so forth. I took Rec-A, a protein. I chose this one for a reason. Rec-A is a universal protein. I like that. Because a universal protein, it is thought, would have to be common to all life, but it would have to be one of the first proteins. So my theory at the beginning of my research was, let's look at the universal proteins. The hypothesis, they will be very simple, and if you get simple proteins, then we can involve more complex ones. Turns out this was anything but. The second reason I like REC-A is because it's about average size for a protein, running around 300 amino acids for an average size protein. So it gives you a ballpark idea just for an average size protein family. What I did was I went to PFAM, downloaded 6,600 sequences. A lot of those are redundant. I wrote some software that would just strip out the redundant stuff, and it turns out there are 4,300 unique REC-A sequences that we have so far. I mean, in reality, you can get, in theory, a massive number, many orders of magnitude more that in theory might be functional as well. But with that, you're able to do a probability distribution for all 20 amino acids at all 326 sites, or 320 in this particular sequence alignment. And it turns out that when you measure the amount of functional information required to code for REC-A, it's 976 functional bits. That is statistically significant by a massive degree. What I'm saying here is that you're looking at a protein that has the fingerprint of intelligence stamped on it. And not just a smudge or a disputable one, but 976 bits is truly impressive. There's different ways you can measure statistical significance, but that is, I would say if you got something that was 50 bits, that is significant. But when you're getting something this high, another way to appreciate that is to look at the probability of getting that through random sampling. The probability in one random sample of getting that would be 10 raised to the 294th power. It's one chance. It's one chance in one with 294 zeros after it. Now, how many actual trials have we had maximum under our standard scenarios? Let's give it 4 billion years since the Earth is supposed to have cooled, and we've got the origin of life and everything else, rapid replication of every 30 minutes, 10 to the 30 life forms. These numbers aren't numbers that pull out of the air. These are numbers that you find in papers that when you draw together to perform these calculations. It turns out that there would be about 10 to the 42, 10 to the 43 trials total. Everything you see on Earth, if you wanted to provide it, if you wanted to explain it using just undirected evolution. For the purpose of this discussion, I'm prepared to grant you any process you want, okay? That's not the issue. What I'm arguing is no matter what process you're going to invoke, you will need intelligence. Not only will you need it, but when you look at the end result, it's got the fingerprints of intelligence all over it. That's showing the functional information. 10 to the 42 trials, massively too small to come up with even one of the universal proteins. It's for this reason. It's this probability problem that Kuhnen says we're going to need an infinite number of universes to explain the origin of life, or the origin of RNA replication at least. What about the simplest life form? You see, I've just talked about one protein here. Simplest life form requires, there are a lot of papers on the Minimal Genome Project, and there's different numbers bandied about. I've just chosen one at 382. Average number of amount of functional information per protein would be about 654. I get that number from 35 protein families I sampled to see what is the average amount of information required to code for the average protein. Some are large, some are small as far as their length goes. Turns out we're talking about 250,000 bits of functional information to encode. You don't really appreciate that until you try and come up with that information by assembling it randomly. One chance in 10 to the 75,000th power. You need a lot of universes to do that, and that's why Kuhnen says you will need almost an infinite number of universes to explain the origin of life. He's an evolutionary biologist, by the way, and not a sympathizer probably with this idea that intelligence was required. I say that probably with a high degree of confidence because that is why you will need an infinite number of universes if there's no intelligence involved in writing the code for life. If you got intelligence doing these things, well, you don't need an infinite number of universes. This actually is not a large amount of information for an intelligent average person in this room to produce. You produce this, you easily can produce this amount of information. We know what can do stuff like this. It's not like we don't know what did that. Therefore, let's see, God did. No, we do know what can produce high degrees of functional information. Intelligence can do stuff like that. And you do it all the time. So we actually have a live candidate on the table. A mind that is intelligent could actually produce computer code, human languages, and the information encoded in the genomes of life. I want to go look at this idea of kinds. Recently, we're noticing that there are more what we call orphan genes than we had anticipated. An orphan gene is a gene that codes for a protein, or at least one protein. Some genes can code for more. But it appears to have little in the way of evolutionary precursors, or none at all. Exactly not what you predict under Darwinian theory. So instead of this idea of climbing Mount Improbable that Richard Dawkins suggested a number of years ago through a series of small steps, when you come to different life forms, like for example humans, in a paper published about, let's see, this is January, published about 4 or 5 months ago, we have at least 60 orphan genes that are unique to us. One would be light years of difference between us and the other primates, but 60. A lot of them have to do, they suspect, with our brain, which would not be surprising. So what we can do, let's say if as a scientist I was interested in this idea, what are these kinds? How would we test for different kinds? I would start looking at the information encoded in the genomes of life, specifically the orphan genes, because the orphan genes would define where the boundaries are between, whether we're talking species, genera, kingdoms, whatever, phyla. We don't know where the kinds are, but when we start looking at the orphan genes, it'll help us begin to make the divisions between the different kinds of life. Okay, so what I'm suggesting here is not only has science not explained how life began, but as science has advanced, particularly in the last 10 years, we are finding positive evidence that intelligence, at least according to some scientists, was required. Now there's a way, if you don't like the conclusion, you have to falsify that hypothesis that I suggested earlier, that functional information, the ability to produce statistically significant levels of it, is a unique property of intelligence. You have to falsify that. There's different ways you can actually test it. You can test it through genetic algorithms, or you can computationally through genetic algorithms. You can also test it in the lab by destroying the information in a gene and seeing how long it takes to evolve back into functionality. And there are experiments going on at the University of Wisconsin for at least a decade now, taking that approach. And very interesting results, very interesting. Bottom line is that in the two ways that we can test that hypothesis, the hypothesis still seems to stand. It's not yet been falsified. And what's more is that there appear to be good reasons why that hypothesis might be true, the more you understand things like genetic algorithms. For those of you who like genetic algorithms, let me just say this. You will know that a key part when you're writing that algorithm, a genetic algorithm, for those of you who don't know anything about it, basically is some computer software you write, or a piece of code, that will try and find an answer to a problem, and it'll use, it's a mix of randomness and some certain amount of guidance. And the guidance is provided by a thing called the fitness function. That can be either encoded within the program, or you can have an external fitness function the program refers to see how close it's getting to the answer. So it's a random search is, let's put on blindfolds and let's, without any sensory perception at all, find a nickel that I left in the Sahara Desert. That's